Okay, so we've been talking about how cells at a high level have systems to regulate genes. And we dived into some of the mechanics of some ways to do that. But now we want to be thinking a lot more as engineers. We are going to say that let's just assume no matter what, whether we're trying to overproduce an endogenous metabolite or introduce some heterologous recombinant pathway, that we are going to have to control expression of some genes. That we're not just and I mean, even if you're trying to overproduce a native metabolite, to do those kind of upregulations and downregulations, you probably also want to do some expression engineering. And so I've called this expression engineering to lump in the idea of gene expression or protein expression, which a lot of people, including me, use often interchangeably. I guess in this course, to be somewhat more precise, gene expression I would think of as being more about your promoter engineering, and protein expression being more about your RBS engineering. And so, again, we're talking about this because fundamentally we want to perturb intracellular enzyme concentrations, and this is how we do that. There are multiple knobs to control. That was one of the key takeaways from last time's lecture. So how do we pick what we want to do? And this idea, I want to throw it out here from the beginning. Is there such a thing as expression that's too strong? And so we'll hopefully come to that, if not this time, next time. We have covered to some degree, but maybe not in as much detail, the cellular machinery that mediates transcription and translation. And then I want to get to a little bit more of the mathematics behind engineering for prediction. I mean, in effect, we've talked about the value of predictive modeling for flux analysis, and we can use entirely different approaches based on biophysics to predict how strong a promoter sequence or RBS sequence might be. And for that portion of this lecture, I'm going to introduce a number of approaches that have really been advanced by Howard Salas, a professor at Penn State, at first his postdoc, and then really since then in his lab. A lot of great work, only some of which I'll talk about. And so all of this will be on focused on bacterial strategies, but we'll make the occasional remarks about eukaryotes. Okay, so I have this here, just even though you're all familiar with it, just to reorient ourselves again. As we're talking about transcription, or the conversion of DNA to RNA, we have this RNA polymerase that's our key molecular actor, and it's what's going to be making single-stranded RNA, which you see here. And actually, it's more than just a circle. You may not be surprised by that. We're going to cover in a sec some of the different components that are involved in it. But right, so in addition to just the circle, we'll also be talking about what the hallmarks of the DNA itself that recruits an RNA polymerase looks like. So if we dive into what these molecular actors of transcription in bacteria are, just mention the RNA polymerase, which consists of some different subunits that we'll describe. Sigma factors are going to be key. They are effectively what mediate the activation of an RNA polymerase on particular promoter regions. So shown here, sigma protein recognizes a promoter, and so you have this RNA polymerase assembly that's already built, and the sigma factor then is sort of that last step required for initiation at a particular region of DNA. And then there's therefore something about that DNA that interacts with the sigma factor. So there are a few different sigma factors, actually more than what are just shown here. And this gets back to the idea also of transcription factors, like we discussed briefly for catabolite repression, that govern a lot of related processes. So understandably, you might want to have, and nature evolved to have, for example, in E. coli and other bacteria, a primary or housekeeping sigma factor. This is just what should be on, generally speaking. And again, just because there's a sigma factor associated with these promoter regions, it doesn't mean actually that the genes are always on. There can be other things downstream that influence whether or not protein is actually made. And so we'll talk about this towards the end of this slide deck, but there are constitutive and inducible promoters. And something to keep in mind here is that regardless of whether it's constitutive or inducible, it still requires a sigma factor. And so you can have then specialized sigma factors for heat shock, for stationary phase, which is effectively one form of nutrient starvation. And then this is another kind of form, nitrogen limitation, having its own sigma factor. We'll spend a little bit of time just talking about the -35 and -10 DNA sequences in these promoter regions, also known as hexamers. And where we're moving towards, if we don't get there by this lecture, we will next, are transcriptional repressors and activators and how their interplay with this promoter region affects whether a gene is on or not, and how you as an engineer might be really interested in trying to manipulate, in particular, these sequences within the promoter region and the characteristics of your transcriptional repressors and activators to control different processes within your cell. And the DNA sequences that the transcriptional repressors and activators bind are known as operators. So far, this has probably been a mouthful of a list of things that some of which you've seen before, some of which maybe you haven't and you're confused about, but we'll dive into that in just a sec. So one slide on eukaryotes, just to illustrate maybe one reason why we're not talking about them as much, is eukaryotes have more factors that influence transcription. And so you have these activator proteins. Well, you have transcriptional activators in bacteria too. But in eukaryotes, there are these enhancer regions that they bind to. And as mentioned here, your enhancers might be thousands of base pairs away. So that's kind of complicated. I mean, you can see if you're trying to control some gene here that you've got to worry about some enhancer that's all the way back here. You've also got to worry about insulators. Insulators can stop enhancers from binding to proteins, and there can be methylation or other kind of epigenetic features that affect whether an insulator or an enhancer are able to control transcription. Okay, so back to our simple systems, like the E. coli RNA polymerase. If we dissect it a little bit, it's made up of these different subunits. We've called out the sigma subunit, and actually it was something I didn't mention is those superscripts, sigma 70 versus 54, they have to do with its molecular weight. And so a lot of the literature about these different RNA polymerase subunits includes numbers that are references to their weights, just as a way of cataloging, and also because I think a lot of the initial experiments that separated and obtained these components used ultracentrifugation techniques that separated components based on their molecular weight. And so we know that the sigma is required for promoter recognition and the initiation of synthesis. Actually, if you have just the rest, and so that's what creates what's called the holo enzyme. But if you have what's called the apo enzyme of the RNA polymerase, just the alpha and beta prime subunits, those are actually capable of, if given a linear single-stranded DNA template, they will also be able to make single-stranded RNA from that. You know, there's some role that the beta is playing in opening and unwinding a double-stranded DNA sequence, but it's really the sigma factor and the holoenzyme complex that is required to both find the right promoter and to start initiation from a double-stranded DNA. And so you can see this architecture at a molecular level, but still represented sort of rectangles and cartoons here with the different regions of the RNA polymerase and the different regions of the promoter. And again, if you're wondering, like, why are we spending this much time talking about promoters? Some of the really cool work in synthetic biology in general, and even in metabolic engineering over the last decade, has been all about changing the DNA in this region, or maybe occasionally a sigma factor, or more likely a transcription factor here, to be able to do innovative sensing and response control of various kinds of enzymes required for production of different metabolites. So that is, again, still the big picture of why we're thinking about this. And I mean, I think when I was a student and at the undergraduate level, I really didn't like trying to memorize what all these different parts of biological systems were and are, just because it felt like endless memorization. But if you think of it more from an engineering lens, like, hey, here are all these parts, almost like preset Legos that nature just gave you, and this is what you can do to change those and to tinker with those to do something really interesting. I think that makes it, that at least gave me a lot more motivation for paying attention to these things. And just to dive in with one more illustration of how a sigma factor might be recruiting the RNA polymerase and what it's doing. This is the sigma factor 54, which was the one involved in nitrogen limitation. And you can see the assembly of the RNA polymerase, in this case, with hexamer regions in the promoter upstream of a gene here. And you can even see that the sigma factor, as illustrated, looks like it has two regions that kind of stick out and associate more with certain parts of the promoter. And you get this activation, ultimately resulting in DNA opening and elongation. And as you have elongation, the sigma factor is no longer needed. So it just kind of pops off.